Welcome to EPG Pathshala. I am Kiran Kumar Salam, working as Assistant Professor in Department of Zoology, Hindu College. Today we will discuss a chapter on molecular genetics, molecular mechanism for regulation of gene expression in eukaryotes. Learning outcomes. First, the purpose of transcription. Second, why initiation in transcription in eukaryote is important and how it is different from that of the prokaryotes. Third, initiating factors involved in the eukaryotic transcription. Fourth, role of the different initiating factors. Fifth, how is straight duplex promoter DNA melted, bent, inserted in the polymerase II active center, enabling the initiation of transcription. Introduction. Transcription is DNA template directed RNA synthesis catalyzed by enzyme DNA dependent RNA polymerases known as RNAPs. It is a universal early step in gene expression because all types of cellular RNA are made by this process. Example, mRNA, tRNA, rRNA, microRNA, small cytoplasmic RNA, etc. The product of transcription is known as a transcript, which gets processed in most cases before becoming functional. Purpose of transcription is helpful in preserving the originality of the DNA. DNA is retained in the nucleus throughout the life of an individual and RNA moves out of it to be used for protein synthesis, that is translation. The process is carried by various RNA polymerases in prokaryotes and eukaryote. In the present chapter, we will discuss the detailed process of transcription initiation in eukaryotes. Initiation is the first step of the process of transcription. For eukaryote, initiation consists of a large number of initiating factors which help in the production of required RNA by using the DNA template. The basic difference which separate the prokaryotes from the eukaryotes are: first, in eukaryote, the process of transcriptions and translations are uncoupled. That is, transcription takes place inside the nucleus, whereas translation site is outside the nucleus in the cytoplasm as shown in the figure 1. However, in prokaryotes, both the processes takes place simultaneously as there is no demarcation of the nuclear membrane. Three different RNA polymerases like polymerase I, 2 and 3 are involved to carry out the process of transcription in eukaryote, which varies according to the type of RNA being produced as shown in the figure 2. RNA polymerase I for synthesizing rRNA, polymerase II for mRNA, polymerase III for tRNA, whereas in the prokaryotes only one RNA polymerase is present. Eukaryotic transcription has regulatory elements such as promoter, enhancer, silencers or terminators. Example, data box located 25 upstream of the start site. The position of such regulatory elements are shown in a figure 3. In eukaryotes, there are several promoter specific transcription factors referred to as the general transcription factors or GTF. Whereas in prokaryotes, a single initiation factor is required, that is the sigma factor. The DNA is present in a coiled as well as the uncoiled form depending on the requirement of the cell, that is the expression of a gene remained precise. Chromatin is composed of the nucleosome, which consists of an octamer of histones around which 147 pairs of DNA are wrapped. The nucleosomal wrapping of DNA plays an inhibitory role in the process of transcription. In the prokaryote, however, the DNA is usually a circular molecule, as we have seen in case of the plasmid. Now, let us discuss a few important scientists and their contribution. Professor Roger D. Kornberg made a remarkable contribution in the eukaryotic transcription. He studied molecular basis of eukaryotic transcription in Stanford University using crystallographic technique. In 2006, he received the Nobel Prize in Chemistry for revealing the transcription apparatus in action, which also helped in analyzing the regulation of eukaryotic transcription. It was known that RNA polymerase II transcribes mRNA using DNA as a template and with the help of several GTFs proteins. So, Kornberg used the yeast to study the role of RNA polymerase II with other proteins GTFs. So, Kornberg used yeast to study the role of RNA polymerase II and other proteins, that is GTFs, during transcription. He made three-dimensional image of the protein cluster using X-ray crystallography and was able to reveal the role of various elements in the transcription. He was able to isolate and identify several proteins participating in the transcription process in their pure form. Thus, he and his research group were able to make several fundamental discoveries concerning the mechanism and regulation of the eukaryotic transcription and convey that these proteins components are remarkably conserved across the eukaryotic kingdom from yeast to human cells. In late 1960s, during his graduation days in Stanford, he discovered the flip-flop and lateral diffusion of phospholipid in lipid bilayer membranes along with Harden-McConnell. In 1970, Kornberg discovered the nucleosome as the basic protein complex packaging chromosomal DNA in the nucleus of the eukaryotic cell. It was Walter Fleming who first discovered that certain structures within the cell nucleus would absorb dye and become visible under the microscope. Kornberg also found that the nucleosome has DNA of around 200 base pairs which is wrapped around the octamer of a histone proteins. However, the major discovery created to these studies was the discovery of the mediator molecule. A mediator is an additional protein complex involved in the transmission of a gene regulatory signals to the RNA polymerase machinery. He has even obtained structural images of RNA polymerases associated with the accessory proteins, which make it possible for us to visualize transcription at a molecular level. The components of the transcription machinery. The transcription process proceeds in an error-free manner and requires the RNA polymerase II enzyme and the general transcription factors, which are referred to as GTFs. Assisted by 5 GTFs, RNA polymerase II in the eukaryotes recognize the start site on a gene, separates the strand of the DNA template, copies one strand of the RNA using ribonucleoside triphosphate as building block, and finally reunites the 2 DNA strands while it translocates along the DNA. The promoter concept was originally derived from the studies of the transcription in the prokaryote, but in the eukaryote they are found to be much more extensive. Studies by Schaffner and Chambon independently in 1981 led to the identification of a gene-specific enhancer elements, that is, DNA sequence that bind gene activator protein which in turn control the transcription of a specific genes. DNA can be seen entering the transcribing complex in a duplex form and unwinding 3 bases before the active site. The 2 strands of DNA are referred to as a template strand and a non-template strands. The RNA polymerase move along the DNA from 3' end towards the 5' end. The template strand reads in the 3' to 5' direction to synthesize RNA in the 5' to 3' direction. You can refer to the diagram given in figure 6. Then the template strand makes a sharp bend, and as a result the next base is flipped pointing down towards the active site. This base is paired with that of the incoming ribonucleotide and the RNA strand. The DNA-RNA hybrid is formed between the growing RNA and the template DNA molecule. The entering DNA is held by a protein jaw within the enzyme. Usually, a wall of the protein blocks the straight passes of the nucleic acid inside the enzyme, leads to the bending of the DNA-RNA hybrid almost at a right angle, with the axis entering the DNA. This results in the exposure of DNA-RNA hybrid for the incorporation of the incoming nucleotide triphosphate or NTPs. These NTPs gain access to the active center via a pore as it comes inside through a funnel-shaped opening. The 5' end of the DNA has an abrupt loop known as the rudder, which prevents the extension of the RNA-DNA hybrid beyond 8 to 10 base pairs. The newly formed RNA passes beneath the rudder and lead, which is another loop of the protein. The lead and the rudder are managed by a large clamp that swings over the active center region in a back and forth manner, retaining the nucleic acid and providing a high level of accuracy. So, important components and their role of the transcription in details. RNA polymerase II is responsible for unwinding the DNA, synthesizing RNA and rewinding the DNA. But polymerase II alone is incapable of recognizing a promoter and initiate a transcription. It requires various other factors called the general transcription factors. General transcription factors. These are required for proper placement of RNA polymerase to the promoter site and to initiate the transcription. Initiating factors are referred to as transcription factors like TF2B, TF2D, TF2E, TF2F and TF2H. TBP, that is Tata binding protein, is a co-promoter recognition in the Tata box. TAF, that is TBP associated factors, is a core promoter recognition in a non-Tata box. TF2A stabilizes the binding of TBP and TAFs. TF2B recruits TF2F and helps in the selection of the start site. TF2F helps RNA polymerase in the binding to the promoter. TF2E recruits and modulates the TF2H, helicase, ATPase and the kinase activities. TF2H helps in unwinding of the promoter through helicase. The detail we will see below. TBP, that is the transcription binding proteins. These are responsible to configure DNA to the RNA polymerase II surface. TBP forms a saddle shaped molecules with the stirrups. A B sheet in the TBP forms the DNA binding site. TBP binds in the minor groove via a strong hydrophobic interaction and a hydrogen bond and induces the large structural changes in the DNA. That is, DNA helices on both the sides form an angle of 110 degrees. TBP bends the promoter DNA around and the polymerase, and the CTD, that is the carboxyl terminal domain of TF2B shown in the figure 6. The NTD, N-terminal domain of the TF2B brings the DNA to a point of the polymerase surface from which it only follows a straight path, and by virtue of the conserved spacing from Tata box to transcription start site in the polymerase II promoters. The start site is juxtaposed with the active center as shown in the figure 6. TF2E enters the complex and recruits TF2H, whose ATPase or helicase subunit introduce negative superhelical tension in the DNA. Mediator molecules. A mediator molecule is a megadalton multiprotein complex which enables the regulation of the transcription. It bridges between the gene activator protein at enhancer and RNA polymerase II at promoters. It helps in the transcription activation and served as a bridge between co-activator and enhancer and between promoter and RNA polymerase. Complexity of eukaryotic transcription, RNA polymerase II core promoters. Promoter help in deciding the point for initiating the process of transcription. Core promoter is a sequence of approximately 40 to 60 nucleotides present either upstream or a downstream the start site and is responsible for accurate initiation. The core promoter sequences are usually flanked by other regulatory sequences known as silencer or enhancers, which bind with regulatory proteins, activators or repressor and modify the rate of transcription by increasing or decreasing it. The various subdivisions of promoters are BRE, that is TF2B recognition element, Tata element or Tata box, initiator, downstream promoter element such as DPE, DCE and MTE. The location of these subdivisions are given in the figure 7. Formation of pre-initiation complex with GTF in eukaryote. The sigma factor which perform the function of site recognition in a bacterial cell is replaced by a set of subunits known as GTFs, which share the homology with the bacterial sigma factor. These GTFs help in the initiation of transcription and melting of DNA. Pre-initiation complex or PIC is association of the RNA polymerase with the GTFs, bound with the promoter site and ready for initiation. Usually, the pre-initiation complex is formed at the Tata element located 30 base pair upstream to the start site and is recognized by TF2D. TF2D denotes transcription factor D for polymerase II. TF2D is a multi-subunit complex and its component which binds to the Tata DNA sequence is known as TBP, that is Tata binding protein. Also, we have TAF, that is TBP associated factors which are responsible for recognizing other core elements, example INR, DPE and DCE. Binding of TF2D at the TBP component distorts the DNA and provides an opportunity for other GTF or polymerase to bind with the promoter. There is an ordered entry of GTFs observed in vitro and the sequences is TF2A, TF2B, TF2F along with polymerase followed by TF2E and TF2H, which are explained in the figure 8. TF2D is the first GTF to bind to the start site and TF2H is the last GTF, and the binding of the TF2H completes the assembly of the transcription initiation complex. The pre-initiation complex containing all the GTFs is followed by the melting of the promoter and requires an ATP which is mediated by TF2H. Transcription factor 2H, that is TF2H, is involved in separating opposing strand of the double-stranded DNA to provide the RNA polymerase access to the single-stranded DNA template. However, only a low or a basal rate of the transcription is driven by pre-initiation complex alone. Transcription factors recognize the promoter, RNA polymerase II then binds and form the pre-initiation complex, which is shown in the figure 9. Other proteins known as activators and repressors along with other associated co-activators and co-repressors are responsible for modulating transcription rate. Activator protein increases the transcription rate, whereas repressor protein decreases the transcription rate. Promoter escape through phosphorylation of the polymerase tail. Promoter escape in the prokaryote is associated by abortive initiation for entering into the elongation phase. But in case of the eukaryote, the promoter escape involves two steps. First, ATP hydrolysis, that is an additional ATP, not the one used for DNA melting. Second, phosphorylation of the polymerase. The carboxyl terminal domain or the CTD of the larger subunit of the polymerase is referred to as the tail and marked by the presence of repeated series of hexapeptide sequence, that is tyrosine, serine, proline, threonine, serine, proline and serine sequence. The repeated presence of this sequence is a highly variable in different organisms. The number of polymerase II CTD varies from 27 in yeast to 32 in Caenorhabditis elegans, 45 in Drosophila and 52 in case of the human, and is usually correlated with the complexity of the genome. Each repeat contained sites for phosphorylation specific for a particular kinase. A large unphosphorylated tail is present in the newly recruited polymerase II, which binds the initiation site. However, polymerase II found during elongation process exhibit the presence of a large number of the phosphate unit in its CTD, that is the tail region. Please refer to figure 10. Phosphorylation helps in the shedding of the various GTFs, and the RNA polymerase can escape or move ahead, leaving behind the promoter. Phosphorylation of the tail region also helps in regulation of elongation process. The role of the CTD in the promoter escape is shown in the figure 10. Role of the TBP. TBP recognizes the minor group of the data element present in the DNA by using the extensive beta sheet of the protein. Usually, DNA recognition involves alpha helix insertion within the major group, which is followed by the alpha factors of the prokaryotes. The main reason for the deviation from the usual pattern is to create a distortion within the localized region in the DNA structure. As we know that the minor group of the DNA is less rich in the chemical information responsible for base pairs identification. The binding of TBP with the DNA results in the widening of the minor group and flattens the DNA by an angle of around 80 degrees. The interaction involves a limited number of hydrogen bonds, and much of the specificity is attributed to the two pairs of phenylalanine side chains intercalating between the base pairs of either end of the recognition sequence, thus creating a strong bend in the DNA. Figure 11 shows how TBP bends the DNA. Next, initiation of the transcription in the eukaryotes and the role of the GTFs and mediators. Unlike the prokaryotic RNA polymerase that can bind to a DNA template on its own, eukaryotes require several other proteins called transcription factors. They first bind to the promoter region and then help recruit appropriate polymerases. The complete assembly of all transcription factors and RNA polymerases binding at the promoter region forms a transcription pre-initiation complex called PIC. Each GTF protein has a specific role in the process of transcription. TF2A binds to the Tata box binding protein, that is TBP, and helps to overcome the repression of transcription. In human, it consists of 3 subunits, namely alpha, beta and gamma. The various functions are, we have interaction with the TBP and facilitates the binding of the TF2D, acts as an anti-depressor by regulating the negative component of TF2D. During active transcription, it acts as a cofactor. It also helps to overcome the rate limiting steps during open complex formation. TF2B is made up of a single polypeptide and has a specific binding site for DNA and TBP within the BRE region. This symmetrical binding leads to the distortion in the rest of the assembly of the pre-initiation complex and helps in the unidirectional transcription. Basically, it acts as a joint between the polymerase and the Tata binding protein, that is the TDP. TF2F binds along with the enzyme polymerase II to the promoter region. This binding of the TF2F provides stability to the DNA TBP TF2B complex before TF2H and TF2E can enter. In human, it is made up of 2 subunits, but in yeast it has 3 subunits. TF2E binds next to the TF2F and has a role in binding and regulating TF2H. In yeast, it consists of 2 subunits. TF2H plays a vital role in converting the pre-initiation complex into the open complex mediated by an ATP dependent transition. In yeast, it's the most complex GTF and consists of around 10 subunits. TAF, that is the Tata binding proteins associated factors, are the 10 in numbers present in promoter region, which actually has INR element and downstream element of the promoter. They are responsible for regulating the binding between TBP and DNA. They show structural homology to histone proteins. Mediators. A mediator is a multi-subunit complex and helps in regulating the expression of RNA polymerase II transcript, which is shown in the figure 12. Sequence-specific DNA binding transcription factors, that is TFs, target these mediator molecules in response to the environmental conditions. Usually, at a basic level, they function by transmitting signals from TFs to the polymerase II enzyme. They help in converting the biological responses to the physiological signals. The need of the mediator arises due to the complex structure of the DNA when it is packed into chromatin. Because of the packaging, the polymerase is unable to bind with the DNA. Activator proteins helps in stabilizing the polymerase binding by recruiting mediator molecules to assist in the DNA and activator binding chromatin modification known as remodeling. They have a broad regulatory influence on the genetic expression, ranging from the chromatin remodeling to the processing of RNA. They are also influenced by the TFs, introns, that is the non-coding region, and the CDK, that is cyclin-dependent kinase 8 molecule. Figure 12 shows how the role of the mediator in formation of the pre-initiation complex. The eukaryotic RNA polymerases or RNAPs. The features of eukaryotic mRNA synthesis are markedly more complex than those of the prokaryotes. Instead of a single polymerase comprising 5 subunits, the eukaryotes have 3 polymerases that are each made up of 10 subunits or more. Each eukaryotic polymerases also requires a distinct set of transcription factors to bring it to the DNA template. RNA polymerase I. It is located in a nucleus, a specialized nuclear substrate in which ribosomal RNA rRNA is transcribed, processed and assembled into ribosomes. The rRNA molecules are considered structural RNAs because they have a cellular role but are not translated into protein. The rRNAs are components of the ribosomes and are essential to the process of translation. RNA polymerase I synthesizes all the rRNAs except for the 5S rRNA molecule. RNA polymerase II. It is located in a nucleus and synthesizes all proteins coding nuclear pre-mRNAs. Eukaryotic pre-mRNAs undergo extensive processing after transcription, but before the translation process, as it is shown in the figure 12. RNA polymerase II is responsible for transcribing the overwhelming majority of the eukaryotic genes, including all of the protein encoding genes, which ultimately are translated into proteins, and genes for the several types of regulatory RNAs, including microRNAs, miRNAs, and long coding RNAs, that is LNC RNAs. RNA polymerase III. It is also located in the nucleus. This polymerase transcribes a variety of structural RNAs. They include the 5S pre-rRNA, pre-tRNAs and the small nuclear pre-RNAs. The tRNAs have a critical role in the translation. They serve as an adapter molecule between the mRNA template and the growing polypeptide chain. Small nuclear RNAs have a variety of functions, including splicing of the pre-mRNAs and regulating the transcription factors. Not all are transcribed by RNA polymerase II, some of them are transcribed by RNA polymerase III also. Let us summarize this chapter. In eukaryote, transcriptions and the translations are not carried on simultaneously. Three different RNA polymerases, namely polymerase 1, 2 and 3 participated to complete the process. Unlike prokaryote, in eukaryote, chromatin is composed of the nucleosomes, which consist of an octamer of histones around which 147 base pairs of DNA are wrapped. Converge shows a detailed crystallographic picture describing the transcription apparatus in full action in an eukaryotic cell. It is now possible to see how new mRNA strand gradually developed and the role of the several other molecules necessary for the transcription process. Regulation of transcription is now easy to understand. Regulatory elements like promoter, enhancer, silencer or terminators, example Tata box located at 25 upstream of the start site, help in genetic expression as the needs of the cell with respect to its environment. Assisted by the 5 general transcription factors, RNA polymerase II in eukaryotes recognizes the start site on the gene, separates the strand of the DNA template, copies one strand into RNA using ribonucleoside triphosphate as a building block, and finally reunites the two DNA strands while it translocates along the DNA. RNA polymerase II is responsible for unwinding DNA, synthesizing RNA and rewinding the DNA. But polymerase II alone is incapable of recognizing the promoter and initiate the transcription. General transcription factors or GTFs are required for proper placement of RNA polymerase to the promoter site and initiate transcription. Initiating factors are referred to as TF, like TF2B, TF2D, TF2E, TF2F and TF2H, each having a specific role in initiation. Pre-initiation complex or a PIC is the association of the RNA polymerase II with GTFs and bound at the promoter site, making it ready for initiation. RNA polymerase II is responsible for transcribing the overwhelming majority of the eukaryotic genes, including all of the proteins encoding genes, which are ultimately translated into proteins, and the genes for several types of the regulatory RNAs, including microRNA and long coding RNAs. Thank you and best of luck.